Good morning. It's wonderful to be here. It's wonderful to kick off this first session. Thank you for the introduction. It's also just wonderful to be back on the conference circuit to see familiar faces, meet friends, and connect with industry peers. So I'm just happy to be here. At MilliporeSigma, over the last 2.5 years or so, we have used the time that COVID has afforded us and taken a little bit of a step back to look at how can we serve the clients in the cell and gene therapy space better. If you're a therapeutic developer, you're probably one of the people that comes to us and says, I need material tomorrow. How can you do this faster? If you're a peer in the CDMO space, you will be well familiar with these comments. And you will be, same as us, trying to speed up these timelines and enable our clients and, as a consequence, patients to get treatments to the market faster. So, as the science and technology company that we are at MilliporeSigma, we have taken 2 steps in that direction. The first one is what you see on the right here. We have revisited our overall structure and identified all the pieces of service that we have in the organization and put them under one roof. So we have combined that into an integrated CDMO business unit, and today I represent the viral vector and mAb space, but we also make ADCs, mRNA, and you may be familiar with our Bauer Alliance testing services that are also part of that business. You can see this now in the market under our new brand, which is Millipore CDMO Services, which covers everything from preclinical all the way to commercial. On the viral vector side, we're very proud to support clients that have commercial CAR-T therapies in the market. I think we have 3 out of the 4 commercial ones that are currently available. And as a consequence, we have been inspected by ANVISA, EMA, PDMA, TGA, and of course the FDA, thus enabling our clients to actually treat patients in these countries. So, what we have also done is, is look at our infrastructure and say, how do we enable that more? What can we do? And so last year we opened our new facility. Traditionally, we have manufactured viral vectors in an adherent manner. Like everyone, we started out with these platforms. We now have the new facility, expanded facility here in Carlsbad, just 2 miles down the road, where we provide up to 1,000-liter suspension cultures, and we cover everything from process development, cell banking, process specific tailoring of processes, and then the large-scale manufacturing in suspension. We continue doing the adherent manufacturing, as most of the CAR-Ts in the space currently still have an adherent platform. So, to get back to speed to the clinic, we have also taken a look at what does a client project look like and how can we make that better? How can we speed things up? Where can we intervene? Where can we find ways of speeding up the different steps of a client project? And so we launched last year our cell line, the medium, and most importantly, the process platform, so that our clients can use our technologies and essentially just say, here's our gene of interest, make it for us as quickly as you can. And so the cell line and the medium sit in our sister business, which is the product business. We have built out the platform, the manufacturing platform, in such a way that, given that viral vector is still a non-stable cell line approach, i.e. we need to transfect the lines at the beginning. But everything else is automated, has been optimized, and so we have been able to reduce 40% of the process development activities because all we need to do is the upstream transfection optimization for the gene of interest. We have the cell lines available, so banking in the initial phase at least is not required. The batch records are pre-written and ready to go. And we have had the first clients go through this with the GMP batch within 9 months. I will also show you that the titers are highly competitive, which is what we're extremely proud of. And we're continuing our R&D efforts to enhance this even further. So if you take a look at the overall Lentiviral platform that we have built out, the process is familiar. You know all of the unit operations, I'm sure. But what we were able to show, is the functional titers are significantly higher than what we have seen in the market so far. We've been able to optimize that as we went along, as we implemented the different improvements across the unit operations. And the step recoveries across different conditions have also been highly improved. So takeaway is we now have several INDs in preparation with our clients that are using this platform. And we have a routine delivery from plasmid handed to us all the way to GMP production within less than 12 months using that lentivirus virus express platform. Likewise, we have just recently launched our AAV offering, which is also cell line that we have developed in-house. It is, again, based on the same routine platforms that you're all familiar with. Upstream needs to be optimized, as I mentioned, for Lenti as well. We focused initially on AAV2 and 5 because those are the ones that are not restricted by IP, but we have since onboarded the first clients in AAV9 and 8, vaulted on 6. And what we see, apologies for the layout. What we see is, again, that we have been able to improve using various process development activities, titers, to ranges that are now highly competitive. I think what we see most in this space is that clients come to us with smaller scale, but everyone wants to go up to 2,000 liters. What I personally see is still limitations on transfection. And so our recommendation at the moment is that we do several 200-liter batches and pool those. But we're looking forward to working with clients and improving the transfection step, maybe looking at different transfection technologies. And then stable cell lines are obviously the overall goal for everyone. So I'm already closing out. Under the new banner of Millipore's CDMO services, we like to approach the market with these 4 taglines. We try to focus the expertise into one business. So I am leveraging a lot of what my new team on the biologics side has to offer, but also what our colleagues on the BioAlliance side have to offer, and we work together on client projects and delivering those together. We have a strong focus on data and digitization, where we not only have these automated batch records and standardization, but also in-process control is a key focus in general, as well as other data and digital platforms. We continue building out our offering through strong R&D efforts. So everything that you have seen with regards to the Virus Express cell line that we have has been based on in-house efforts. And I think we're all very, very proud of the R&D teams that we have that have delivered this in record time. And then at Merck in general, or MilliporeSigma in general, sustainability is becoming a bigger topic with the sister business in the product space, with the bioreactors, media, and others. We're working together to obviously reduce the impact on the planet and everything that we do, and continue focusing on that. So with that, I have a little bit of time for questions. If there are any, but okay. Thank you.